The overall goal of the following experiment is to determine whether changes in mutation frequencies with increased replicative age of yeast cells are due simply to additional rounds of cell division or to age-specific changes in rates of accumulating mutations. This is achieved by first measuring mutation frequencies and rates in young cells by spreading cells onto selective and non-selective media to calculate a predicted mutation frequency for cells of increasing ages due to mutation accumulation with each round of cell division. Next, a yeast cell population is labeled with biotin, grown in liquid culture, and the original labeled cells are recovered by magnetic sorting to obtain aged mother cells that have undergone several cell divisions. Then, aged mother cells are either regrown to increase their age and then sorted again, or stained for butt scars, and then spread onto selective and non-selective media in order to determine their average replicative age and mutation frequency. Results are obtained that show whether there are any age-specific increases or decreases in accumulating mutations based on a comparison of the experimentally observed mutation frequency to the predicted mutation frequency for cells of the observed average age.
This method can help answer key questions in the field of aging, such as, do changes in the rates of accumulating genetic damage contribute to normal aging? Or are the lifespan-altering effects of various genetic and environmental factors partly due to changes in mutation rates? Demonstrating the procedure will be Melissa Patterson, a graduate student in my laboratory.